My name is Sweta Ravenor, and I'm a senior from San Jose, California. Currently, microarray experiments are some of the most widely used assays for gene expression-based studies. They basically tell us how a gene behaves in different tissue samples. And data from more than 14,000 microarray platforms has been made publicly available. So in a nutshell, I used a high-performance computing cluster to analyze nearly 1 billion public domain gene expression data points in 2 phases. In phase 1, I constructed an unsupervised machine learning algorithm to generate detection calls that tell us if a transcript of a particular gene is present or not. And for each gene, I used its expression data from all samples, expectation maximization techniques, and Bayesian statistics to generate a Gaussian mixture model. So each gene is basically represented as a curve that looks like this. And somewhere along that frequency distribution, there lies a threshold for gene presence. To dynamically calculate this gene-specific threshold, I harness the likelihood ratio, which is used frequently in clinical trials. So in samples to the left of this threshold, the gene is not present, and in samples on or to the right, the gene is present. And when I tested my algorithm against existing software, I found that it identified present genes with a 97% agreement rate. And unlike existing software, my algorithm is not reliant on platform-specific information, meaning that it's workable across all microarray platforms and species. One of the biggest advantages of having this pan-platform microarray algorithm is that it paves the way for precision medicine. Detection call signatures can be used to assign clinical phenotypes to tissue samples, something like low-grade versus high-grade, and from here, patient subgroups can be defined for personalized or precision treatment. During my background reading, I also came across another issue. Experimental methods for predicting gene function are labor-intensive and error-prone. So in phase 2, I extended my project to predict the biological functions for genes based on their mixture models. I verified my predicted gene functions using gene ontology enrichment, pathway overrepresentation analysis, existing databases, and literature, and I found that my workflow correctly predicted 67 essential genes in mice and nearly 4,000 immune genes in both mice and humans. The identification of essential genes is a crucial step in the development of new drugs, whether we're talking about antifungal pesticides or human medications. And on the other hand, immune response genes have been shown to be effective biomarkers for several diseases. The rapid prediction of these new essential and immune genes in phase 2 allows us to expand the drug development target space and systematically identify biomarkers. So we have phase 1, which is an accurate and comprehensive detection call algorithm, and phase 2, which is essential and immune gene prediction. And ultimately, my project showcases the faculty of bioinformatics as a whole.